Welcome to another episode of Tiny Talks Big Ideas podcast. In this series, we shall explore the scientific papers that literally changed the world.
Correct.
Okay, take a deep breath with me. Feel that sort of energy humming inside you, the basic fact of being alive. We usually picture evolution as, this fierce survival of the fittest battle, but what if? What if the biggest jumps forward in life weren't about fighting at all, but about radical teamwork?
Exactly. Think about this. Those tiny power plants working away inside almost every single one of your cells right now, the mitochondria, they might not actually be you originally.
Not me. What do you mean?
Well, the idea is they could be ancient guests, maybe even invaders, sounds a bit dramatic, but organisms that set up shop inside our distant ancestors billions of years back. Permanent roommates.
The invaders inside us definitely has that sci-fi vibe you mentioned.
It really does. But the amazing thing is it's looking like profound biological truth, a truth that kind of rewires how we think about ourselves, about what we even means at a cellular level.
And like a lot of truly revolutionary ideas, this one faced some serious pushback. For ages, it was pretty much seen as scientific heresy, wasn't it?
Oh, absolutely. Dismissed, ridiculed even.
So today, we're diving into that story. The story of Lynn Sagan, who later became the incredibly famous Lynn Margulis, and her really audacious 1967 paper on the origin of mitosing cells. This deep dive is all about how her groundbreaking theory totally flipped our understanding of evolution and life itself, revealing these hidden ancient communities like thriving inside us. To really get how radical this was, we need to rewind a bit. Picture the mid-1960s. Biology basically saw life divided into two huge empires. Huge gap between them.
Right. On one side, you had the prokaryotes. These are your bacteria, blue-green algae, really simple cells.
Like the studio apartment of the cellular world. Tiny, tough, no frills, just a loop of DNA floating about, no nucleus, none of that complex internal stuff.
Exactly. The survival machines, and then the eukaryotes, animals, plants, fungi, us. The complex cells, the mansion cells, you could say.
Right, with a dedicated nucleus for the DNA, the mitochondria churning out energy, maybe chloroplasts if you're a plant.
And scientists back then saw this huge divide. They literally called it the greatest single evolutionary discontinuity, a massive chasm. And the big question, the real head-scratcher, was how do you get from that simple studio apartment to the complex country estate? Especially since there were no obvious stepping stones, no missing links found.
But here's the thing. The clues were kind of sitting right there, weren't they? Hiding in plain sight, you might say. Inside those complex eukaryotic cells, structures like mitochondria and chloroplasts were acting, well, weirdly.
Very weirdly. They had their own DNA, for starters, and it was circular, like bacterial DNA, not linear like the DNA in the cell's nucleus.
And they reproduced on their own schedule, right? Just sort of splitting in two, like bacteria do. They even looked a bit like bacteria under the microscope.
Precisely. It was like finding these tiny, independent little organisms living inside the main cell. But the dominant idea, the accepted wisdom, was that all this complexity must have evolved slowly, internally, step by step.
So the idea of one cell just engulfing another and setting up a permanent partnership, a symbiotic takeover.
It just seemed, outrageous, fantastic even. It didn't fit the narrative of gradual internal change that everyone was working with.
Enter Lynn Sagan. Later Margulis, a brilliant, really tenacious 28-year-old scientist at Boston University back then, fresh off her PhD.
And she had this hunch, more than a hunch, really, a deep conviction built on synthesizing older, almost forgotten ideas about symbiosis with the latest biochemical findings. She saw connections others were missing.
She wasn't just pulling it out of thin air. She was connecting dots across different fields.
Absolutely, drawing on cytology, biochemistry, paleontology. And she argued that symbiosis, this living together, wasn't just some minor evolutionary sideshow. It was a major driver.
But getting that idea out there, wow. Her 1967 paper on the origin of mitosing cells faced a real uphill battle, rejected by something like 15 journals.
That's the story. It was dense, packed with detail across disciplines, but at its heart was this incredibly radical narrative about where complex cells came from.
And that core idea was endosymbiosis. Let's break that down. Endo meaning within, and symbiosis meaning living together.
So literally one organism living permanently inside another. Not slow renovation, she argued, but more like a corporate merger, a joining of forces.
Okay, let's paint that picture. 2 billion years ago, maybe. You've got a large, relatively simple host cell. Anaerobic oxygen is poison to it, and the atmosphere is starting to fill with oxygen. Big problem.
Right. Meanwhile, you've got these smaller bacteria that have evolved this neat trick. They can use oxygen, not just tolerate it, but use it to generate massive amounts of energy. They're thriving in this new oxygen-rich world.
So the big anaerobic cell engulfs one of these smaller aerobic guys, maybe initially for food, who knows?
Right.
But then something different happens.
Instead of digestion, a partnership forms. The little aerobic bacterium finds a safe haven inside the bigger cell, protected from predators or whatever, and in return,
It becomes an internal power pack, pumping out energy using that dangerous oxygen, but safely contained inside. The host gets energy, the guest gets protection.
and that, Margulis proposed, is the origin of the mitochondrion, the powerhouse of virtually all complex cells today. It wasn't just adding a feature, it was a fundamental transformation, making complex aerobic life possible.
Wow. Okay, so that's merger number one, but she didn't stop there.
Nope. Later, she argued, some of these early eukaryotes, already equipped with mitochondria, did it again. They engulfed another type of prokaryote this time, one that could photosynthesize, like cyanobacteria or blue-green algae.
The ones that use sunlight for energy.
Exactly, and those became chloroplasts, the little solar panels inside plant and algae cells, providing basically free food from sunlight. So now you potentially have three formerly independent life forms merged into one cooperative unit.
Mind blow. Okay. And then there was a third hypothesis, even more debated at the time, involving movement.
Yes, the idea that flagella and cilia, those whip-like tails or hair-like structures some cells use to move, might have originated from symbiotic, free-swimming bacteria, maybe something like spirochetes.
So they brought motility to the partnership.
Initially, perhaps, but Margulis took it further. She suggested these structures, these former bacteria, were later co-opted for a completely different, absolutely crucial job, managing cell division, mitosis.
Like,
The idea is that the organizing centers for mitosis, things like centrioles and the structures that pull chromosomes apart, might have evolved from these motile symbionts. They became the machinery ensuring that when a complex cell divided, the DNA was shared out perfectly. An incredibly complex dance, choreographed by ancient partners.
so this whole picture,
Yeah.
it's not just about where organelles came from. It fundamentally changes how we think about evolution itself, doesn't it?
Absolutely. It shifts the focus. Evolution isn't just about competition, red in tooth and claw. It's also about collaboration, about synergy, about building something radically new by joining forces. It implies that a cell isn't really a single thing. It's a community, a chimera.
A walking, talking ecosystem, essentially.
You got it. And crucially, this wasn't just a nice story. Margulis laid it out. This is testable. If mitochondria and chloroplasts were once free-living bacteria, they should still carry the evidence, ghosts of their past.
Their own DNA, their own way of making proteins, their own method of reproduction.
Exactly. She basically predicted what molecular biologists should find if they looked closely enough.
But the initial reaction from the scientific community was less than enthusiastic.
It's putting it mildly. It was largely hostile, dismissed, ignored, even ridiculed. It flew in the face of the established dogma of slow, gradual, internal evolution. It just seemed too out there.
For decades, it remained on the fringes, right? Seen as too radical, maybe unprovable.
Pretty much. The tools just weren't quite there yet to really dig into the molecular details she predicted. The community wasn't ready for cells as ancient communities.
Until the 1970s and 80s. Technology catches up. Molecular biology comes of age.
And boom, the smoking gun started appearing, just as she'd predicted. They found mitochondrial DNA distinct from the nuclear DNA, circular like bacterial DNA,
Just like she said.
and mitochondrial ribosomes, the protein-making factories. They look bacterial, not eukaryotic. And crucially, direct observation confirmed. Mitochondria and chloroplasts reproduce by binary fission, splitting in two like bacteria, independent of the host cell's division cycle.
They weren't being built by the cell, they were self-replicating residents, passed down generation after generation, mostly through the maternal line.
Exactly. And then, definitive genetic evidence in 1978 from Schwartz and Dayhoff nailed the chloroplast connection directly to cyanobacteria. The pieces just started falling into place, overwhelmingly supporting her core ideas.
So this once fringe theory, it moved to the center. It became fundamental, textbook biology. It completely redrew the map of the cell. And recognition followed, like her election to the National Academy of Sciences in 1983.
The impact was just immense. For evolutionary biology, it showed how complexity could leap forward through partnership. For medicine, understanding mitochondrial diseases suddenly made more sense. These are issues with semi-autonomous entities within our cells. Think about certain cancers, Parkinson's.
Even affects things like agriculture, right? Understanding chloroplasts for crop engineering.
Definitely. And just philosophically, it shifted our view of life. Less purely competitive, more interconnected. Our cells aren't single entities. They're bustling metropolises, these intricate ecosystems.
And Lynn Margulis, she continued to champion and refine these ideas throughout her incredible career, right? Became a distinguished professor, received the National Medal of Science, the Darwin Wallace Medal.
An absolute giant in the field. Her later work expanded on these themes. She passed away in 2011, leaving this huge legacy.
There's often talk about her deserving a Nobel Prize.
There is, and many feel strongly she should have received one. But regardless, her true legacy is embedded in how we teach and understand biology today. She showed us evolution's cooperative side, its radical creativity through partnership. Plus, she was a prolific writer, speaker, managed research, teaching, family. Truly remarkable.
And incredibly, the story keeps evolving, doesn't it? We're still figuring out the details of this ancient partnership. How do the host cell and these organelles talk to each other?
It's an ongoing conversation, billions of years old, happening inside you right now. And new discoveries keep reinforcing our framework. Just recently, in 2024, scientists identified what seems to be the first nitroplast, an organelle derived from a nitrogen-fixing bacterium living inside an alga.
Wow, so these kinds of mergers might still be happening, or at least we're finding more examples.
Exactly. It suggests this symbiotic pathway to complexity is a recurring theme in evolution. It also helps explain some long-standing puzzles, like why the range of DNA-based compositions is relatively narrow in eukaryotes compared to the huge diversity in prokaryotes, consistent with a more sudden symbiotic origin rather than purely gradual divergence.
Which really brings us back to those big questions for you listening right now. Are you truly an individual? Or are you this incredibly complex ancient community? Could other big evolutionary leaps have happened this way? Is symbiosis, this coming together, maybe the secret engine behind life's amazing creativity? So yeah, you're not just you, you're fundamentally a we, powered by ancient roommates, a whole universe cooperating inside every single cell. Until next time we meet in another episode of Tiny Talks Big Ideas podcast, stay safe, stay healthy, and stay curious.